Russell Galanti is a junior extension agent for ornamental crops and landscape industries. He is a graduate of CTAHR and has worked in the green industry for 17 years. Russell works with the cut flower, nursery crop, turf grass, and landscaping industries through research and educational programs. His presentation today is research updates from Oahu and Hawaii Island ornamental extension projects. Russell, thank you for joining us.
Thanks, Glenn. So update, that was 3 years ago. It's now 20 years I've been working in the green industry. So I need to give you an updated one. Hello, humans behind the camera. I know you're there, even though I can't see you because I've been moderating. So I have an idea of who's here also, so I'll try to cater the talk to those individuals. I'm going to talk today about all of the work that I've been doing as the ornamental extension agent for Oahu and Hawaii counties. I'm going to focus mainly on the red ginger production, and I'm going to talk a little bit about my Protea project. And then I'm going to give a little bit of a plug to come to the field day tomorrow to take a look at our sunflower trial as well as our eucalyptus trial. So as I said, I'm going to talk a lot about viruses affecting red ginger because this is one of the largest projects I've been dealing with as an extension agent starting on Oahu County. But this project's statewide, and I do want to give thanks to Adriana Larrea Sarmiento, a PhD student in the lab of Dr. John Hu. She's done a bulk of the work. She's done all of the lab work for this project. I've had the good fortune of being the person that flies around the state and finds these viruses and characterizes them and meets all the cool growers, sees all the cool plants, but ultimately is the person that's characterizing them from the farm perspective. The lab of Dr. John Hu are the individuals who are doing the real meat and bones of the work. So they're doing the work before high throughput sequencing, which is that HTS up there. High throughput sequencing is a method for essentially rapidly and cost effectively sequencing DNA and RNA, pretty much the way that we determine what viruses are which viruses at this point. And the image you're seeing here is just to give you an example of the sheer amount of work they've been doing. It doesn't look like a lot, but it's a lot of lab work that goes into it. We pretty much made it through number 1, number 2, virus detection, virus selection, and characterization of these viruses. And so I'm at the point today where we're going to discuss what we found out and then where we're going after that. So if you know me or know anything about this, you have an understanding of the problem we're dealing with. For many years, specifically starting on Oahu, multiple viruses have been identified as causal agents for decline symptoms in Alpinia purpurata. That's the red ginger, and Alpinia purpurata cultivars. So that's your Kimmy Pink, Jungle King, Jungle Queen, Raspberry, etc. If they're Alpinia purpurata cultivars, they're most likely affected by these viruses. We've also found other plants in the Zingiber family that have issues with some of the viruses that we've determined. And in fact, these viruses are problems in many different plants, as I'm going to show you. So we've identified a bunch of viruses, and there's a possibility for additional viruses as well. One of the things that I've done specifically in collaboration with Adriana is characterize the symptoms. And we've come up with N number of symptoms. Specifically for me, stunting, so I'm going to go through by the letters A, B, and C. Stunting, dark and light banding on immature stems and on mature stems are some of the most common symptoms that we've seen. Some images of that are right here. They're really hard to see on some samples because they're very light and not dramatic in terms of the symptoms, even though this dark and light banding is the most common symptom we've seen in association with viruses. And if you're concerned with me not telling you which viruses are associated with these symptoms, I'm going to get to that in a few minutes. This is also in the field very symptomatic of what this dark and light banding is. And this is associated with a new virus that we've identified. It's not just new to gingers, it's new to science. It has no name. We have no idea what else it infects. Nobody else has identified this virus in the world. So we're at the point where we're identifying new species. Other symptoms include bleached chlorosis, leaf deformation, chlorotic mottling, and those look a little something like this. Now, this is associated with another new virus that we've determined, completely new to science again, new to the world. It's our new worst friend, best enemy. So you're seeing the chlorotic bleaching. It looks like either sun bleaching or possibly, in a lot of cases, this is what herbicide damage looks like. So if you were to spray your plants with glyphosate on accident, you'd also see similar symptoms. And we've seen some similarities between glyphosate damage on ginger and this new virus B, this unnamed virus. Beyond that, we've got some of the more typical symptoms that were identified in the beginning of this, well, I'll call it a pandemic. And those were mottling patterns, chlorotic mosaicing, marginal leaf yellowing, leaf necrosis, stem dieback, and low production of stems. So that's G or F through K, along with L and marginal leaf bleaching. These were the typical symptoms that we were seeing when we first were identifying viruses. So this was before my time. This was 3 years and more during virus identification. And these images are all indicative of those symptoms. So on the top left, you've got some of the mottling. Moving over to the top right, the mosaic patterns, marginal leaf yellowing on the top left, and some marginal leaf necrosis, and then a whole suite of combinations in between, including the spray brush yellowing and chlorosis. So we knew of some viruses that were already infecting these gingers, and now this is work that was done before myself. BBRMV was one virus that was identified that is affecting gingers. That's banana bract mosaic virus, BBRMV. Now, this is a serious virus for these plants, but as we go through this talk, you're going to realize, as I realize, that even though it's serious, if your plant is infected with it, it's not a virus that is infecting a lot of plants. So it's not prevalent in the plants throughout the state. So it's not one that we're seriously concerned about because it's low level of spread. Now, canny yellow mottle virus is a virus that we are pretty concerned about because it is fairly prevalent and it does have symptoms and effects that are associated with the decline symptoms. So that's CAYMV, canny yellow mottle virus. And you can guess where that originally was found, in cannas. So once we start to get into the virus issues and realize that it's a virus complex, we have an understanding that these viruses can infect the plants, co-infect the plants. So we've got cross-infections with viruses, banana bract mosaic virus and canny yellow mottle virus. This is another serious issue for plants because both of these viruses in one plant are associated with decline symptoms. But once again, BBRMV isn't found commonly as a virus that's infecting plants, so it's not something that we're seriously concerned about. And then we've got canny yellow mottle virus in association with BCMV, bean common mosaic virus. Now this is another co-infection that was discovered, and it's a mixed co-infection. These two also cause severe decline symptoms, although they're slightly different from the others. You can see the difference in these symptoms. So we have more leaf necrosis and the mosaicing pattern on the edges for the BBRMV and canny yellow mottle virus. I'm going to slip up a lot because there's a lot of tongue twisters. And you see more of a mosaicing pattern in the CAYMV and the bean common mosaic virus mixed positive. So that's reviewing what we've already identified before this project. And we do know already we've got these 4 viruses. They can all be mixed together. And out of all of them, banana bract mosaic virus and canny yellow mottle virus are some of the more serious issues because they are more commonly found than bean common mosaic virus and another one, banana streak virus, which we're basically not worried about at all. Now, there was some consideration that the ginger decline could be associated with a fungal pathogen or possibly even a bacteria pathogen. We have biologists on the project. We also have mycologists and bacteriologists. Bacteria pathogens have pretty much been completely ruled out. That's not to say that there aren't bacterial pathogens in gingers, but they are not associated with these decline symptoms. We haven't been able to find a single bacterial pathogen that has been associated on plants that we've been sampling. Now, fungal pathogens, as Lisa Keith was telling you, are everywhere. And there are a ton of different fungal pathogens that affect gingers. But we've also determined that they're not necessarily implicated in the decline of this red ginger. Now, we did determine one fungal pathogen that was prevalent in all of the samples that we collected that had the symptoms that you're looking at up here. So that's these yellow leaf splotches, and that fungal pathogen is Nigrospora. We didn't identify the species because the mycologists couldn't identify them. We also were not able to perform Koch's postulates. So Lisa Keith also went over how to perform Koch's postulates. And we weren't able to transmit Nigrospora from an infected sample and plate it and then put it onto a non-infected sample. Most likely because the plants that we were using were too healthy. Although Nigrospora is seen throughout ginger production. You've most likely, if you're a ginger producer, you've most likely seen these symptoms, which I'm gonna forward to what these symptoms look like in the field. Thank you, Hannah Lutjen, for the sample on the right-hand side. On the left-hand side is a sample from Oahu. So while you might think you have virus in your ginger, if you're looking at symptoms like this, you most likely just have a fungal pathogen, and that pathogen is most likely Nigrospora species. Now, there are some issues there because it has been shown through our sampling that sometimes plants that are infected with viruses are more susceptible to the Nigrospora species. So you will see more fungal pathogens infecting plants that have been infected with these viruses, although it's not always the case. So this is the breakdown of what we've determined so far. And you can see virus A and virus B are at the bottom. There are unnamed viruses. We have an idea of what they are, but we're being fairly secretive about it because the virologists want to publish, so we can't necessarily give all the juicy details away about these viruses. So the column on the very right is showing prevalence. We've taken 80 samples. I had to be a little bit conservative with how many samples I took throughout the state because it takes a long time to do the sequencing for these samples. So for example, I went to Maui in, I think it was May, and we just got the samples sequenced a week ago for these samples. So you can imagine it takes a long time to do this work, especially with COVID. Always the excuse of not being able to get supplies, et cetera, shipping. So I really like this table because it gives you an idea of how many of these samples had each virus associated with them. So canny yellow mottle virus, you can see it's fairly prevalent. Over 50% of the samples, just over 50% of the samples tested positive for canny yellow mottle virus. Only 8 tested positive for banana bract mosaic virus. So if I'm correct, that's what, 10%? None tested positive for bean common mosaic virus. So we're throwing that one right out. It's almost non-relevant in the virome project that we're working on. Banana streak virus, BSV, also pretty unrelevant because only about 3 samples have been tested positive. Now, virus A is our biggest concern right now. 52 samples tested positive for virus A. Along with virus B, 36 samples tested positive for virus B, and that's out of 80 samples, yeah? Let me catch up on my other computer. I'm breaking the numbers down a little bit different right here because some of you are interested in quarantine and whether or not we should be shipping plants between islands. I'm not here to say you should or shouldn't. I'm here to give you the information to determine whether or not that's going to be something useful. And there are some patterns that we see here. We're starting with Kauai first. I traveled to Kauai once. Thank you, James Keach, for also helping me around Kauai and for all the individuals who let me sample at their farms. We only identified virus A and candy yellow mottle virus on Kauai. So we can't say for certainty that the other viruses aren't there. In fact, they most likely are, but they're not there in high quantities, especially in the farms that we were sampling and at the nurseries that we were sampling, some of those nurseries that ship statewide. Now Oahu's just a hotbed. It's a veritable petri dish. We identified most of the viruses there in my study. We also identified bean common mosaic virus there before the study that I'm discussing right now. Of course, virus A, again, is the most prevalent. Virus B is the most prevalent on Oahu. In fact, virus B is a huge issue on Oahu itself. Most of the images that you see from virus B are from Oahu. Let's see. BBRMV, banana bract mosaic virus, fairly short, small amount are identified on Oahu, and banana streak virus, pretty much negligible. Maui, again, we only found virus A and candy yellow mottle virus. So if you're a grower on Maui and you're interested in knowing what the results were, I've got some good news for some of you and some bad news for some of you. So you can contact me and I'll contact you about what plants tested positive and what plants tested negative. Big Island, another petri dish, just like Oahu. Most likely plants are being shipped from Big Island to Oahu, so you're seeing some of those viruses being shipped with those plants. So banana bract mosaic virus, virus A, virus B, candy yellow mottle virus, and banana streak virus were all identified on Big Island as well. And these are showing the number of samples that tested positive for each virus. So, is it getting any clearer or is it getting more complicated? It's only gotten more complicated for me in some ways, and it's gotten more clear in other ways. And it's still something that's a gray area and somewhat nebulous. But I have teased out some general speculations from this project. So banana streak virus, bean common mosaic virus, and banana bract mosaic virus, they're the drama queens of, or drama kings, drama humans of the virus symptoms here. They create dramatic virus symptoms, right? And then when they're a combination with each other, they create incredibly dramatic symptoms. But for some reason, maybe it's that they're not as easily spread. Let's go back to here. Most likely due to their persistence, banana bract mosaic virus and bean common mosaic virus are not spread as easily because they're non-persistent in the insect host. So they go away. An insect sucks on that plant. The aphids most likely are feeding on the plants. They have the virus on them, but if they don't feed quick enough on another plant, they're not spreading the viruses. And the two other viruses that we're dealing with are only semi-persistent. So they may not be spreading the viruses fast enough, or the viruses aren't viral enough to spread to the plants. But nonetheless, they create incredibly dramatic symptoms, and stunted growth as well as stem dieback and full plant decline to plant death are associated with single infections of each single virus as well as cross infections between these viruses. But once again, they're not very prevalent, so I'm not as concerned about these viruses in the industry because their inability to spread rapidly. They're not the COVID of the plant viruses that are affecting gingers. Now, virus A is a virus of interest and concern because of its high prevalence throughout the state. It was found on every island. It was nearly always the most prevalent virus in plant samples. we're doing on time. Here's some images of what virus A looks like. You probably noticed that these are the same images that I showed you for the streaking patterns originally for the symptoms, because virus A by itself almost only ever shows these symptoms. The plants can look fully healthy, they can be producing perfectly and at high production, but they will be infected with virus A. So this is kind of the good news in my opinion. Virus A by itself, especially with healthy plants, does not seem to have a major effect on production, at least with age. The plants aren't, or with young age, the plants aren't infected. That's not to say that virus A won't eventually become too high of a viral load and infect the plants to such a degree that they go into decline symptoms. So I'm not necessarily trying to advocate for not dealing with a virus if you have virus A. I'm just saying that the plants can be productive and somewhat healthy if you only have virus A. For example, here's what the virus looks like in super healthy fields. On the left-hand side, you've got a pretty much healthy plant just with the banding patterns. And on the right, we've got a field that is infected with virus A. Some of the plants are, but the plants themselves are very healthy and they're very productive. So I'm going to kind of wax on this, that plant health is going to be your best defense if you've got viral load in your fields already. But once virus A co-infects with any other viruses, you start to see really dramatic decline symptoms. And this is going to be the case no matter what. If you've got a co-infection of viruses, you're going to have decline symptoms. So on the left-hand side, we've got virus A, virus B, and canny yellow mottle virus. You can make an assumption about where this plant was found. On the right-hand side, we've got virus A and canny yellow mottle virus. So unfortunately, if you've got virus A and canny yellow mottle virus, you're most likely going to have severe decline symptoms. And then any other virus A and other virus combination is going to lead to decline symptoms. Virus B. I love just getting to call them virus A and virus B. You have typical streaking. So sometimes plants will only show the banding patterns. But virus B is unique in that it introduced these new symptoms that we had not seen before. Bleached, distorted foliage that looks very similar to herbicide damage. Here's what they look like in the field. So almost always the bleached, distorted foliage is going to be on the very newest growth. Now, I've seen fully bleached and distorted plants that are producing very large flowers. They just aren't very tall normally because the distorted foliage kind of stunts the plant, but it will produce a fully whole and large flower. And here is another interesting symptom that we've just discovered in a virus B only infected plant. So this plant wasn't showing necessarily that typical bleached, distorted foliage, but it is showing some symptoms, including stuntedness, stem dieback, and just in general low production, which is not a symptom we necessarily were characterizing, but one that is associated with co-infections as well as virus B itself. Clear as mud. So general conclusions to make it a little bit more simplified. And this is not just what I assume, this is after having meetings with the virologist and determining that this is what we thought is going to be a general idea of what the virome issue is. So ginger plants presenting co-infection with the new virus A, virus B, in combination with canny yellow mottle virus are associated with the decline of the plants. So if you got virus A, you're going to be pretty much okay. Now, that's not to say always. If the plants are unhealthy or if there's something else infecting the plants, you're going to have some decline. If you have virus B or if you have virus A in combination with canny yellow mottle virus or any of the other viruses, you're going to have an issue most likely with decline of your plants. Infections of BBRMV, and I'll add on to this BSV and BCMV, along with any of the other above viruses, will induce severe decline symptoms. And I added this myself. Starting virus-free and focusing on plant health are going to be the best options for productive plants, because I myself am charged with trying to find the best solution for dealing with these viruses. One more note on the virus work. Oh boy, 5 minutes, ladies and gentlemen. Vector transmission. We're doing vector transmission experiments to try to see if virus A and virus B are being vectored by mealybugs, scale insects, and aphids. So far, we've been unsuccessful. We're going to wait a little bit longer and see how symptoms develop on plants that we stuck with these viruses. Another plug, plant health, in my opinion, plays a role in how the plants express viral load. So all three of these images are of plants that are infected with virus A only. The plant on the left, it receives no water. It's in complete drought. It only gets rainfall, and it's in a fairly drought-stricken area. Those plants are incredibly declined. They're showing stuntedness, low production. They're showing all of the symptomatic symptoms for the decline. The middle image, and these are not great examples because they're not right next to each other necessarily. They're not the same distance apart, but take my word for it. The middle image is of plants that receive some irrigation and rainfall, and they are slightly healthier. And then, of course, on the right-hand side, we've got plants that are in high organic matter. They're in a high rainfall area, and despite getting fungal pathogens more, they are more healthy, and they are seeming to produce more. So it would be interesting to infect some plants with viruses and then treat them with neglect and then treat them really well and see how they react to being infected with the viruses. For that, we need more money. Virus-free production. This is what I'm going to focus on before I focus on anything else, because I want to get virus-free plants out to the industry. We're looking to produce virus-free plants. We're looking for locations to grow them, and we're looking for funding to do tissue culture work with Hawaii Ag Research Center. Here's a picture of me producing or creating a virus-free insect exclusion enclosure. Here's some virus-free plants that we've discovered. Thanks, Hannah, for the virus-free plants we found on Maui. We got Kimmy Pink, Jungle King, and we got 4 red gingers from Maui, as well as red gingers from Kauai. I'm going to have to skip the Protea production because we're out of time.
Yes. Sorry, Russell. I mean,
I should have asked for 45 minutes.
Oh, you guys should have swapped. Yes.
Yeah.
Okay, so just a quick question here from one of the viewers. It seems most of the virus vectors are caused by insect pests. Does this mean effective virus prevention is highly correlated with routine use of insecticides? And is there an effective organic method for pest control?
Routine use of pesticides and organic use of pest control. You know, that's a good question, but I'm not an entomologist, so I'm not going to be able to answer that very well. I was recommending use of pesticides to eliminate insect pests that can spread the virus. I'm going back also. We might want to switch back over to the image that I'm about to show you that shows whether or not mechanical transmission can infect these viruses also. There we go. So you can see whether or not mechanical transmission, so that's whether or not you use your pruning shears to cut your plants, whether or not that will spread the pathogens. Okay. So it's going to be impossible to prevent insect pests from entering ginger fields. So while you can spray, you're never going to spray enough to keep mealybugs and aphids out of your fields. I've got growers that spray weekly, and they're unable to have mealybugs completely eradicated from their fields. And, plug. I've got a publication, sorry, Glenn. I've got a publication that outlines all of the pesticides that can be used in ginger production. So if you want to email me or call me, I'll put my contact information in the chat. You can get that publication from me. So you can If you're interested in using organic production, I've got some options. If you're interested in using chemicals, I've got options. Thanks, I'm gone.
Thank you, Russell.